Hello, my name is Antonio Politi. I work in the facility for light microscopy at the Max Planck Institute for Multidisciplinary Sciences in Göttingen. In our facility, we support users in their image acquisition and in their data analysis, but also support them for workflows where they need feedback microscopy. So what we try to do is combine different ingredients. We have microscopes, obviously, and they could be different. They can be a wide field microscope, a laser scanning microscope, or low for super resolution, like STED microscope, or doing even different tasks for fluorescence recovery after photobleaching. We may also want to combine other devices, like microfluidic devices. And another additive feedback microscopy workflow is also combining processing tools, which is some image analysis task, and then in a workflow manager, put everything together so that we interleave image acquisition and analysis and changes in image acquisition during the workflow. Sometimes we may have an API, which is disclosed by a company, that allow us to directly control the microscopy software.
So in the different scenario, how this can happen, they are basically in one case where we have a microscopy software which controls the microscope, provides also workflow management and some image analysis tools. And with this, within this framework, we can do a lot of things already. But if there is a little bit of code or tools that are available, we may outsource some of the task. So being the first task we may want to outsource is the image analysis, to be able to do maybe more complex tasks or reuse the workflow that we already programmed and you would like to use it again and use it also on different microscopes. It can be more and more that we even manage the workflow, so the microscope software is only needed to basically start the different imaging sessions, or having something where nearly everything is controlled by the API. It's still in the framework, it's still the microscope software. So we're not controlling the single parts or the single mirrors, but we can send comments and all the settings for the different acquisition can be described in the API and set in the API. So I typically like to work also as Alex more in this range where in fact we use a lot of the tools that are already in the microscope software, which gives some advantages because maybe the users can choose the settings, the appropriate settings for the imaging. I don't have to program it somehow ad hoc, and you can choose, you know, this low laser, medium laser, high laser, which some heuristic that they think is important. And then maybe in the other part of the software, we just switch between the different imaging settings depending on some decisions made by image analysis. So in my opinion, not all problems are good for additive feedback microscopy. And there are certain prerequisites you want, and also to discuss with the users. And as a facility, we also discuss with our users and propose, okay, is this an interesting solution for you or is it not? And do you want to acquire the data as you normally do? So, and I think, first of all, you want to acquire medium to large data acquisition. So you want to acquire enough samples in a kind of standardized way. So your samples, your type of samples you must have enough. So there are certain projects where your samples is very precious. So we have here project that run on human tissue, live, maybe some oocytes that there are once in a month you get one. And this you will not run with this kind of workflow because it's just too precious that maybe there's some bug or something not working. So you says, okay, the user will sit in front of a microscope and run the experiments to get the best possible data they can. And also there is a kind of sweet point for me, what I found, what measurement should require. So if the measurement requires a few minutes to 1 hour, 1 or 2 hours, then it's a good example because you may think in an overnight experiments, then I can run many, many of these measurements. If my experiments anyway runs 8 hours, then I can come the next day, switch my sample and continue. So there's no point of having such additive feedback microscopy experiments. And ideally, basically also the two goals in this is for this kind of experiments is to achieve, of course, something that a high amount of data, but also to best use the instruments. So if you can run the instruments day and night, that's the goal, so that for the time that they are in use, we really make the best out of them. So, and the type of experiments, you identify rare cellular states. I work also a lot with live, but also a little bit with fixed samples, and automatically optimize imaging area. Then sometimes you overcome some intrinsic limitation of microscopy software by some tricks using the software you design, and maybe mix different experiments. So FCS, FRAP, super resolution, and some exchange of fluidics in between. And the audience, yes, that's really a good question. So who is always what I mentioned a few times during the today, who is the expert? Or I think, I mean, I work in a facility, so I like how facilities are conceived. So that basically you can discuss with the scientists, and they're willing also to optimize the acquisition workflow in order to achieve this higher throughput. So, and this takes a few steps. So it doesn't work maybe from the beginning, but then at the end, they get really a nice workflow where they can achieve a lot of data that they, if this is the project that they want to achieve. If of course they need only 3 examples, images, and it's so clear the phenotype, they may not need this effort. And ideally, you try to joint effort with an expert, whatever this expert means. Somebody that has done this before and can implement maybe some new features, I would say in this case. So for data analysis, what kind of problems are we addressing? And there's nothing special. So it's nothing, but it can be very problem specific. So I think, classic problem is that you have colors in the cells and staying in different colors, or maybe in transmission, and you want to do an instance segmentation. So identify the different objects. You may also want to do some tracking. And so these cells in the middle is in fact in tracked. So all the other cells are running around it and moving around it. But the microscope is basically keeping these cells in the middle, because we want to see when it changes state from this classification where we classify the S phase with this marker. And then when all these little dots go away, and we'll go to a G2 state. And we were interested in this case to identify exactly this time and then wait for 2 hours and then start the imaging. So there was kind of a very ad hoc experiments for a certain project where we needed a little bit of tracking, segmentation, classification on top of it. Sometimes we also do geometry. So, and this is, I have the impression many image analysis problems are geometry, you know, finding the longest filaments, the longest neurons with the biggest bud at the end, or the top of the nucleus or the bottom of the nucleus for doing some imaging there, or some data extraction there. So, and from algorithm I use from rather simple algorithm to more complicated algorithm, but there's no preference saying, okay, we want it should be deep learning, or because that's the more fancy one. Whatever works. And in fact, some of these algorithms may take a lot of training data, and maybe in fact an overkill for certain problems. So you maybe well do thresholding a watershed, you have done and you solve the problem. And this is much faster. You don't need to create training data sets. You don't need to worry about you will still have errors, but comparable to the CNN errors. So the problems goes from 5D, so we can have 2-dimensional, 3-dimensional, where we have to use multicolors, or also time and integrate over time what to get an information to make a decision. And it could be part of a feedback experiment. So as we described, identify target to trigger different mode of acquisition. It can be something that I rarely do, but I think would be really useful, because when you're starting creating particularly large data sets, depending on the microscope system you have, so a spinning disk or a light sheet microscope that creates a lot of data sets very quickly, then it would be nice to provide also some feedback to the users where we are standing, and spare time, because if the processing all the data takes a day and the acquisition take a few hours, you may want to really parallelize your analysis during the acquisition to be faster and get the results much faster. So, and this We do a little bit, but not, in fact, not enough, where this during the feedback experiment, during acquisition, you can either process the data on the acquisition computer, but this can be sometimes limiting because is not the best hardware after a while. You know, maybe your hardware is 2 to 3 years old, and you may want to go to more powerful dedicated machines, and we use virtual machines for this. So, and then of course, every of this kind of workflows create a lot of data and show the data analysis. We want to be quantitative and extract eventually some numbers out of it or some biological information out of it. So the data analysis workflow should be intrinsic part of these experiments afterwards. So I would like now to give you 3 examples of what I've been doing. And I'm happy that at least I couldn't listen to the first talk, but so I will give one example of super resolution microscopy, which is using an Aberria and Nikon setup. And then 2 examples which run on a little bit older now Zeiss platform, which is for doing automated FCS and FRAP. So we also work on other systems, which are not only in the facility, but also in other departments. And they often have now a Python API. And basically the API means they disclose some of the commands that you can execute in the GUI. They are also in a Python format so that we can run these commands and the parameter for these commands for the image acquisition in a Python language and call them either within the software or from outside by loading the DLL of it. So STED is a super resolution method that allows to basically, when you normally, so it's a laser scanning method, and you will normally go with a confocal beam and excite several fluorophores in the excitation beam, but due to the use of a depletion beam, which is surrounding more or less the excitation beam, you deplete or inactivate many of the fluorophores. So that at the end you get an effectively smaller point-spec function and a super resolved image. So why it is good to do feedback microscopy on STED is that STED is nice. So we have a 2D STED, which is a Steadicon from Aberria. So it's a nice method because it's super resolution of fixed and live samples. It's confocal, but it's really slow because you want to achieve quite a lot of photons out of here, but you remove 90% of the photons. You have to record very slowly for every pixel. So you have a very long pixel dwell time. And so this can take, depending on the size of the image, 1 minutes to 30 minutes per image or image stack. So, and then you have to use quite high laser power. So you bleach and photo damage a lot. So you can acquire maybe 1 image or 2 images per sample, and you have to move to the next one. And also because due to the speed, your field of view is quite small. So this is kind of the bad side of this method. So we have another method, which is in fact very fast and gentle, which is wide field microscope. It's not confocal, but for many problems, it's okay. And obviously it doesn't have any super resolution capabilities, but it really can bridge what we cannot do here. So using a camera and it's relatively cheap setup. So we have now very sensitive cameras, and with tiny bit of light, you can get very large tiled image of many cells and decide which cells to image. So, and then we decided to couple these 2 systems. So, and the Steadicon system is a relatively simple system. So from what the company decided to make a system that's very user-friendly. So basically it's like there's a button to say, start the image, and it acquires an image. And you can put some settings on it, some laser, STED laser and excitation laser, but there's not a lot you can choose, which is good also from this side. But they have one feature that you could basically put the system in waiting mode and then trigger an acquisition that you previously decided how to do it with just a TTL signal. So basically this workflow works that you first decide, okay, which are the best settings for my current experiment on the normal software that is provided by the company, and then put it in waiting mode. So on the other side, we have a Nikon standard software, which is called NS Elements. And they also have a workflow manager, which is quite nice, which is called Jobs, where you can control the workflow and the stage camera and decide also some image analysis task. To this, we wrote extra code in a macro language to also be able to talk to an external program, which in this case is ImageJ, but it could be anything. And this was very simplistic because we couldn't figure out something better, honestly, to write in and out some coordinates in a file and listen to when this coordinates changes. So, and then the workflows runs, then here it records either tile image or several positions, then send these images to ImageJ. ImageJ decided what to do with it, segment it, whatever we want, and then send back yes or no coordinates. And then when these coordinates, then the stage move there to the new coordinates and says, okay, send a triggering signal to the Steadicon system. So you see that basically we are not doing anything special to the system, but suddenly we are able to do from a system that came without not even multiposition. Now it has changed, but in the original version, there was no multiposition, to a system where we can do a lot of acquisition. So the data analysis was very simple. So from such kind of image, you segment and find cells, and then we wanted to image at different positions depending on the questions at the top, bottom, and middle. And these were one of the reasons why we use ImageJ, because in the software from Nikon software, we couldn't really find a good algorithm to do it. And then image respective positions instead. And then there's obviously some post-processing where the biological question came in. For instance, this was more of a proof of principle examples, and we look at nuclear pores. So, and we were able to image in an overnight run, which was for 12 hours. So about slightly less than 1,000 cells. And this is really nice because normally you would, for every images, you will acquire 1 to 3 minutes, and doing this manually, it can be really, really boring and very tedious. So, and with this, we could then try to answer questions, biological questions that requires, in fact, a lot of statistics. So, for instance, we asked, okay, what happens if you put different inhibitors of the cell cycles? How does the pore density changes? And what affects the new pores generations? And this is, we could measure with the pipeline, we could measure how the pore density, how many number of pores per square microns, and then check for different drugs, even very small changes, because this was in fact only 2 experiments for one of our students. And you could imagine you can do much more than this very easily, a third overnight experiments to increase the number of samples. And then also identify very tiny changes in the composition of these nuclear pores in interface cells. For instance, finding whether for this component and one of our reference component, where there is a difference. So where there's one component is where they're always present both, or one component is more abundant than the other component. And this, for the biology, was interesting because it tells us how the pores are assembled when the cells grows. And we are looking at small percentage changes, so 2 to 3% changes, which are really hard to pick with small numbers of cells. So that was interesting to show that using this automated super resolution method, we can achieve enough numbers to come over the noise that is intrinsic to our data and get some biological results out of it. So my next 2 examples is more on the classic laser scanning microscope system. And still, when I was also at the EMBL, I developed a tool that allows me to combine different image analysis workflows or image analysis platforms and perform some rather standard, what we call now standard experiments, follow cells, do some FRAP experiments or FCS experiments, or detection of rare events. And then in one effort, we we combined FCS, which is a method to basically measures the fluctuations of fluorophore passing through the confocal volume. And from this fluctuation, you can read out the number of molecules, diffusion properties, and if you are in two colors, even protein-protein interactions. And this method is also one of the methods that is rather tedious to do it manually, because you need a lot of statistics, but the measurement time is rather slow. So you do one experiment, takes 1 to 2 minutes, and then you have to move your stage, go to the next cells, place some points, measure for 1 to 2 minutes, and do the next one and next one. This is extremely boring. And if you do this for 4 hours, which is your time you're on the microscope, you really get very depressed from it. So, and that's where we use quite a lot this automated workflow to basically do this kind of experiments. So we combine a few things together. And one was to do what we call FCS calibrated imaging, which is basically running an experiment where we identify cells automatically classify cells in an overview image that are entering mitosis. And then these cells are then imaged at high resolution in 3D. So we have a 4D images for these cells. And at the end of the measurements, we automatically acquire FCS measurements. So, and this is here a segmentation and classification problem. And here at the end, it's basically a simple segmentation problem, and then geometrically deciding, okay, where is the best positions to place my FCS points. And from this, in post-processing, calculate the concentration and calibrate the images. So converting the fluorescence data into concentrations. So what we were able to do it for many cells that have been transfected endogenously with fluorescence protein to basically acquire in a very standardized way data throughout cell divisions, starting from prophase up to late anaphase, and have this in a quantitative way. And the data was so quantitative and so standardized that we were able then also to create kind of an average distributions of this protein. So this is shown for 3 proteins, but for metaphase up to anaphase, we were able to derive for every of this protein kind of the average distribution throughout. So the next example, you can also use this to do experiments where you want to probe the molecular biophysical properties of your proteins through time by basically repeating these FCS measurements in a regular distance. And for instance, this is a protein that during mitosis built up a complex, so diffusion coefficients reduces and becomes smaller. So it means it goes into larger complexes, and then after the end of anaphase, this protein is released and is then freely diffusible again. Another problems that we solved with a similar setup was to be able to do multi-position FRAP. And this is something that, at least on the system we have, is not possible to do. Basically, we want to achieve a time resolution below 5 minutes and have enough cells, and we can only do FRAP in one field of view. So using the cell size we have, the density we want to have, and the objective we have for this kind of measurements, we could measure 5 or maximally 10 cells in a field of view. So ideally, we would like to do it in several positions and only record the area which we are interested in. So, and then this is the reason why we use the same tools to do a multi-position FRAP experiment. So where, again, finding an overview image, finding cells that are interesting, define position and define some bleach ROI, and basically put them in a list of things we want to do. So, and this list, we say, okay, about 20 to 25, so enough cells that we could image them within 5 minutes time interval. So when we have found all the cells, then the protocol starts and it goes to every position, does the FRAP and does the normal FRAP experiments and record the recovery for over 5 hours. And then with this, we had to combine all the post-acquisition analysis, because having this data is pretty useless if you cannot analyze it, and basically extracting a camograph and tracking a camograph around the cells, aligning then the camograph and finding the regions that corresponds to the unbleached and bleached region. And with these, we were able to automatically extract traces for every of these experiments, and then compute it for many cells, and then now for many different cell types. So we are looking at something like 20 to 30 different cell types, and this is currently running this kind of experiments where we're running day and night and try to produce enough data now for a publication for it. So which are our future plans? I think has been already mentioned. So one part where our institute is well known is super resolution, and in fact, one of the recent development in our institute was MinFlux, which is a very powerful super resolution method and tracking method, but it's also very slow. So and you can acquire only to get enough sampling, you acquire a rather small area, and we would like to have this in multicolor. So we already have paint working multicolor. So paint is a way of labeling your protein by, for instance, using a DNA homology and labeling different imaging strands and different homology strands, labeling different proteins and targets in your samples. This already works with this technology, but the experiment to do two-color experiments is basically 5 hours sitting there and then washing several times because we want to image several regions. So, and this is something which would be really nice to be able to run it overnight, also because these instruments are, because they are so slow in acquiring data, they are used basically all the time. So during the day, the time is full. So we try to use also the night time as much as possible so that we get data out of it. And they're also very expensive, unfortunately. So the second part is more on the data analysis part. And basically also here, and what I work in the past was a lot of recognizing cells that are going through mitosis. However, in our institute, we are working a lot in meiosis, and this has many more phases than, in fact, mitosis. There's all the prophase, where there's all the chromosome coupling and crossover, which is very interesting. And some groups are able now to visualize this and microscopy this for quite a long time. And basically, we will try to build classification regression models to recognize cells in the different cellular stages, and also to do different experiments with it. Maybe some high temporal resolution images to look how the nuclei are moving during this this kind of growth of oocytes, and also do some photoactivation or cutting experiments. So, and this is more on the, we're collecting data from the different collaboration groups and try really to build something generic for not just for mitosis, but for all these development problems. So going from egg to first embryonic division, and then build to use all these tools on dedicated virtual machines. There can be accessible from different microscopes and can not only be used from the microscope in the facility, but also from the microscope in the different departments of our institutes if they need it and have problems to address there. So my last slides sounds maybe negative slides, but I think this has been named a few times, which are the limitations and bottlenecks for me and for many of us. And so what are the things where we can work well with it, but where we feel a little bit limited? So integrated solutions, I think they're great. Nikon had, in our case, a very nice integrated solutions, but we were lucky there were some scripting capabilities, but some of their image analysis tools just didn't work out at the end as we expected. And also cost. So we can buy one license, we can buy a second license, but then it gets really expensive to buy many licenses to develop code on it and to develop tools on it. So we are super happy if there is an API And many companies now provide this kind of API. And however, it often lacks documentation and examples. And this is really, we need examples. Example is even better than documentation. Give me an example how to do A and B, and this I understand what's going on. So, and sometimes depending on the company, can be either very old type of language or maybe a typical language syntax, and it can be really cumbersome to develop on it. So, and depending on the company size, feature request is hard or impossible. Fun enough, small companies are more willing to implement features than larger companies. Larger companies have much bigger cycles, and it may not be an interest to develop a feature, whereas smaller companies are like, they may say, oh, that's a good idea, let's try it, and in the next version you have it. So they may have much faster cycles. Support, yeah, sometimes, and this also depends on the company side, sometimes only very few persons know the details of the workflow manager or API. So when we're using these solutions, then we have either to help ourselves, but if you're trying to ask, they try the best. I think I found really people that help us, but there are not so many. And then, yeah, they cannot do miracles. So they help if they can. So, and I think we should put as a community an effort to sharing the tools, where and how to link it. I think also defining what we need, as has been named a few times. Maybe how can we bring, if we're developing tools for a certain microscope or for a certain platform, how can we make it visible also for other people using these microscopes? And maybe some companies provide a platform where this can be uploaded with examples. I mean, there's a lot of possibilities for this. With this, I think I would like to finish.